Recording. Great, we are now recording. Thank you very much for agreeing to give a Build a Cell seminar, and it's all yours.
Okay, then I will share my screen. Can you see it?
Yep, we can see your presentation.
So yes, I want to briefly introduce myself. I am Eleonora Bailoni, a PhD candidate in the University of Groningen in the Netherlands. You might know the Netherlands for its many beautiful flowers and tulips, for the many bicycles and the windmills. And yeah, here is where I am doing my PhD. And my main project is coupling, cofactor regeneration, and phospholipid biosynthesis. So today I will present what I've been doing so far. But I want to start by saying a few words about the BASIC consortium that I am part of. So the BASIC consortium is a national Dutch consortium that is focused on building a synthetic cell from the bottom up. So we want to exploit non-living components to create an autonomous self-reproducing synthetical minimal cell that hopefully has lifelike properties. And the aim to do so, the goal for doing so is to hopefully learn a lot about living system on the way of getting there. So we divide the work in working packages, and I am part of working package 1 that is focused on self-fueling. So my talk will be about a minimal metabolism and really focused on fueling. But on top of that, there are other working packages, such as the DNA processing working package, the cell division working package, and all of these working packages are working on a specific point or part of the synthetic cell. And later on, these will be integrated together by further working packages. So, but to build on the minimal metabolism that is required for self-fueling, I want to explain that what we need is a compartment that can host membrane proteins. And these are really essential for the controlled import and export of nutrients and of waste products. So the synthetic cell should be an open system, but a selectively open system, so that not all the metabolites can flow in and out in an uncontrolled fashion. And on top of that, we should also include a system for internal recycling of those byproducts that for some reason cannot be removed out of the synthetic cell, but that if left there would slowly build up in the internal lumen and cause inhibition. So this is the base to develop a sustainable minimal metabolism. And once we look a bit further into what modules we need for that, we definitely need to achieve physical chemical homeostasis for the synthetic cell. And that implies that we have a very good control over pH as well as over volume. And on top of that, we need to be able to achieve homeostasis of the nucleotides. So the ATP that is required for all anabolic reactions, for example. We do need a system for the synthesis of phospholipids that are essential in order to expand the membrane, which is a key point before cellular growth and division can happen. And importantly, we also need a system to assist membrane protein insertion because, well, as I've already stressed, that membrane proteins are an essential aspect of the synthetic cell, but they do not naturally come to the lipid bilayer. They need to be inserted by a sec translocon, for example. And in addition to that, we also need components to import all the essential building blocks for growth and division, and that is amino acid for protein synthesis, nucleotides for DNA duplication, and so on. And once again, I want to stress out that the minimal metabolism that we design needs to be built in a way such that the byproducts and the final products have to leave the system and the substrates have to come from the external medium. And of course, this is a very ambitious goal. Many people are working on it in my university, and each of us is really focused on one of these modules. But importantly, we are now trying to combine them to build complexity and to hopefully achieve a more sophisticated network of reactions toward minimal metabolism. So today I will talk about my own project that is related to energy homeostasis and how we tried to couple it to phospholipid biosynthesis. So I will present the work that I have done together with the phospholipid expert Martin Exterkate in order to bring phospholipid biosynthesis to the lumen of the synthetic cells and make it sustainable over time. So I want to start with a little bit of a background on a pathway that Martin had developed previously for continuous phospholipid biosynthesis from fatty acids. So this is a synthetic pathway that takes a fatty acid molecule. It activates it by means of ATP into an acyl-CoA active unit that is then attached to the head group glycerol 3-phosphate. And when two acyl chains are attached, phosphatidic acid is formed. This is the simplest form of a phospholipid that doesn't have any functionalization on the head group. Now, from the phosphatidic acid, we can proceed. There is a formation of an intermediate, CDP-DAG. And from CDP-DAG, he reconstituted the pathway in a branch fashion. On one side, he could form phosphatidyl ethanolamine, and on the other side, he could form phosphatidyl glycerol. And these two types of phospholipids are very relevant from a physiological perspective because together they might make up the majority of typical membranes, such as the Escherichia coli membrane bilayer. And so the system is very tunable, also by changing the fatty acid composition. He showed that he could produce various types of phospholipids. So not only the head group, but also the acyl chain could be changed. And what is important is that the synthesis is really complete. Conversion is to 100%. So it is shown in this graph over here. As a control, when only oleic acid was added, but no enzymes, or no enzymes from either of the branch, then PE or PG synthesis was not observed. However, when he added PGSA and PGPA for PG synthesis, he could achieve a complete formation of PG with a very little bit of PE. Similarly, when PSSA, serine, and PSD were added, so all the components for PE synthesis were present, then PE was synthesized with only a very little bit of PG. But when the most interesting and intriguing aspect of the pathway is that when both the proteins for PE formation and for PG formation, when mixed together, then different ratios of PG over PE could be achieved. And this varied based on the relative amounts of enzymes for PG formation and for PG formation on the right and for PE formation on the left. And what is most interesting is that in the last condition, number 6, he managed to achieve 30% PG synthesis and 70% PE synthesis, thereby coming very close to the composition of an Escherichia coli lipid bilayer. So then the question that we asked together was, how can we make this pathway sustainable for our minimal metabolism for the synthetic cell? And you can ask this question. It is very fair to ask because only very few of these substrates are actually sustainable. CoA is taken up by the first enzyme and attached to the fatty acid to form acyl-CoA. Once acyl-CoA is used up, the CoA is released. So CoA is recycled by the pathway. And similarly to that, carbon dioxide, a byproduct that is formed in phosphatidyl ethanolamine formation, is released by passive diffusion to the membrane bilayer. However, there are many more components that are not sustainable. ATP needs to be fed from the inside of the synthetic cells. Same for the head group, glycerol 3-phosphate, because this is not membrane permeable substrate. So we either would need to have an importer or to indeed synthesize it from the inside. And the same goes for CTP. So really my talk today will be about how we tried and achieved nucleotide homeostasis and namely ATP and CTP recycling. And then how we also tackled the synthesis of glycerol 3-phosphate. So I want to start by showing you how we coupled CTP synthesis with glycerol 3-phosphate formation. For that, we decided to work with a somewhat simpler setup. So as you see in the picture above, we didn't use the whole pathway for phospholipid biosynthesis, but rather we started from a phospholipid already, phosphatidic acid, and we looked at the functionalization of its head group towards phosphatidyl glycerol formation. And because this branch of the pathway requires CTP and glycerol 3-phosphate as well, we added two components. The first component is a module for CTP generation. So we achieved that by starting from cetidyl monophosphate and adding two soluble enzymes that are called CMK and NDK. And these enzymes, they phosphorylate the cetidyl monophosphate in two consecutive reactions into cetidyl 3-phosphate. And they do so by taking up a total of two ATP molecules per CMP that is converted into CTP. And on top of that, we also included a step for glycerol 3-phosphate formation, and that required the use of glycerol kinase, an enzyme that phosphorylates glycerols also by using ATP. And what is really the advantage of this reaction is that glycerol is membrane permeable, so we can just feed glycerol from the external medium. We do not need an importer for glycerol. So here are a few results. When we have the phospholipids, but none of the components, we are basically looking at our substrate levels. So the total PA that is present in our system. And then we prepare a positive control that contains CTP and glycerol 3-phosphate. So really, this is only a test that the phospholipid biosynthesis pathway is working. And we observe PG built up. So quite a significant synthesis of PG. Next, we add CMP. But not yet any of the enzymes for CMP phosphorylation. So obviously we lose the PG signal here because synthesis cannot happen from CMP. However, when we do add both CMK and NDK, we restore the signal for PG because now we have enzymatically produced CTP in the system that allows for the reaction to proceed. As a next step, we want to include glycerol 3-phosphate formation, but we don't do that right away. We first take a negative control Well, we add glycerol, but without the enzyme. So there is no glycerol kinase in this reaction here. And what you observe is then that there is an accumulation of the first intermediate CDP-DAG. Now, this is expected because if you look at the pathway, the first enzyme, CDSA, doesn't require glycerol 3-phosphate. Glycerol 3-phosphate is only required by the second enzyme, PGSA. So the first reaction takes place because CTP is there. However, CDP-DAG cannot be further converted into PG. However, when we finally add glycerol kinase as well, we observe G buildup, and this is comparable to our positive control in condition number 2. So we are very pleased with these results. And now you might ask, why is synthesis not complete? Why do we, in all instances, still have significant amounts of phosphatidic acid? Our interpretation of this data is that synthesis is happening at one side of the lipid bilayer, and therefore only half of the PA that is present in the membranes is actually available for conversion. So this is a very important result in our research. And as you might have noticed, now all the reactions that are presented here, so CTP generation and glycerol 3-phosphate formation, are in function of ATP. So obviously we need to have a very efficient module or pathway for the generation of ATP inside the synthetic cells. And for that, we decided to use the arginine deiminase pathway that has been developed in our laboratory previously. This is a fairly simple pathway that is only composed of 3 soluble enzymes, ARC-A, ARC-B, and ARC-C, plus a membrane antiporter, ARC-D. And the beauty of this pathway is that it takes up one arginine molecule from the external medium, and through a few enzymatic steps, it converts it into one ornithine molecule that is exported back by the very same enzyme that imports the substrate. So you can already appreciate here that the system is out of equilibrium in that the substrates can be fed from the outside, and the byproducts are also not accumulated inside the lumen. And then in the last enzymatic step, ADP is converted into ATP. So we follow that over time, or it was done before me, by looking at the ratio of ATP over ADP as is reported in this graph. And I want you to focus on the black line, which is a line in a condition where pH was kept constant by addition of an ionophore. This was important for us because the sensor for ATP relative to ADP is pH sensitive. But what you can appreciate is that when arginine is added at time point 0, there is a buildup of ATP relative to ADP over time, and this signal is constant at least within 2 to 3 hours. Now, why do I say that the pH has to be constant? Because, well, it has to be constant, but why does it change? It changes because the pathway produces 1 ammonium ion for the last reaction and 1 ammonium ion in the first reaction. And these ammonium ions, as you can better see from the figure in the middle, they are in an equilibrium. So the ammonium ion will equilibrate with a proton and ammonia. And ammonia, because it is neutral, will diffuse out according to its concentration gradient and leave the proton behind. So this proton really contributes to the acidification of the lumen of our synthetic cells. But really the take-home message from this slide is that there is a pathway for ATP generation that is sustainable because the substrate is taken up from the medium, the byproducts are either exported or they spontaneously leave the system by passive diffusion. So what we did was to combine this pathway with the first enzyme towards phospholipid biosynthesis, which is glycerol kinase I've shown before. So now we performed a very similar experiment, and here we are again looking at the ratio of ATP over ADP. And when we start the pathway with arginine addition, we do see a very rapid increase in or build-up of ATP relative to ADP in the absence of glycerol. However, the build-up is not so prominent in the presence of glycerol, and this is due to the fact that glycerol kinase is rapidly hydrolyzing ATP to form glycerol 3-phosphate. So this type of measurement really gives a feeling that the ATP is being recycled and that glycerol 3-phosphate is being synthesized. And really, this is the base for further optimization and implementation of the phospholipid biosynthesis enzymes inside the synthetic cell. But now I want to come back to this slide that I've shown already, because I want to say a few more words over fatty acids. And I've colored them in red and blue because ideally the feed of fatty acids is not problematic. It is very well known that they can equilibrate across the bilayer. However, we encountered some issues, so I want to further elaborate on this. So yeah, this is a very simple experiment that I have performed. However, these data sets are also present in the literature already. So just as a small recap for those who might not know, oleic acid or oleic acid or any other type of fatty acid is present in solution in a neutral form and in a negative form. And the relative ratio of these two forms is based on the pH. So based on the pH, there will be a different relative population of the two forms. And the neutral form can equilibrate across the lipid bilayer very, very rapidly. This is also what I observe here in an experiment where I prepare vesicles with a pH probe on the inside, so phospholipid vesicles. And once I add oleic acid, I see an immediate pH drop that is due to the neutral form immediately equilibrating to the other side of the bilayer, thereby releasing a proton. And this proton release causes an internal acidification in the lumen of the vesicle. And then the second mechanism is diffusion of the negative form of oleic acid, and this is much slower because, of course, the negative charge doesn't want to diffuse so well through the lipid bilayer. However, and you can see that this happens much slower. So in the time scale of minutes, you see a recovery in the pH. This is due to the negative form of oleic acid diffusing and taking up a proton. And when the system is fully equilibrated, then the pH is back to the initial pH. So I also borrowed this picture on the right from a paper that I really suggest reading. I can recommend it because I found it very interesting. Here, a small molecule uncoupling protein mimic was developed. So it is this, well, pink drawing, but the chemical structure is available here. And what this molecule does is it shields the negative charge of the negative oleic acid. And in this way, it increases the equilibration rate of the negative form of oleic acid. So this is really valuable work. I think if we would find out that the flip-flop of oleic acid would be limiting in our setup. But this is not the end of the story. And I want to build on that by reminding you that oleic acid is very poorly soluble. Its critical micellar concentration is perhaps a few micromolar, depending on how it was measured. And what happens above the critical micellar concentration is that it will rearrange into supramolecular structures based on the pH. So about neutral pH, we find so-called cubosomes. When the pH is increased, the oleic acid and other fatty acids as well, they reassemble into vesicles when about they're half in the neutral state and half in the negative state. And from there on, we proceed towards micellar state when most of the fatty acids are in the negative state. And this really caused a problem for us because, well, first of all, we observed aggregates when we tried to dissolve oleic acid at a neutral pH. And so we had to work at higher pH, at around pH 8, to obtain a more homogeneous solution. But again, I want to point out that this is a very turbid solution, probably containing oleic acid vesicles or assemblies of oleic acid that are not in the free form. This is really important when you think of feeding this oleic acid to the phospholipid vesicles, because ideally you want to feed enough that the vesicles can really duplicate their membrane surface. But for that, the concentration that is needed is really quite significant. And what can happen is that by feeding concentrated oleic acid, you are feeding oleic acid vesicles instead of free monomers of the fatty acid. When a vesicle of oleic acid might come into contact with a phospholipid vesicle, then there might be some leakage which we want to avoid. So this was a really important control for us to measure the leakage of our phospholipid vesicles. And indeed, we saw that we obtained a very significant leakage in the presence of oleic acid concentrations that are relevant for our synthesis purposes. So really the take-home message here is that yes, oleic acid can diffuse through the membrane bilayer when it is in the free form, but when it is more concentrated, then it can cause leakage, and therefore we concluded that we couldn't perform phospholipid biosynthesis in bulk, but rather we had to develop a technique that would allow us to feed diluted oleic acid over a prolonged period of time. And that brings me to my second goal, or to our second goal in our shared project, which was to develop a system to control the external environment of the synthetic cells in order to ensure a constant substrate supply and also to prevent the buildup of waste products in the external medium of the synthetic cells. So to better explain what we had in mind, I have here a picture of our sustainable minimal metabolism that is placed into closed compartments, so a test tube. And now what we wanted to do was to develop a technique or a technology where the vesicles could rather be placed into an open compartment. And by that, I mean a compartment where the vesicles could be separated by a dialysis filter and be in contact to always be in contact with a fresh feed of substrate so that the vesicles could be retained, but the substrate could always flow and equilibrate across the dialysis filter into the vesicle chamber, so that similarly the waste products produced by the vesicles could be washed away through the feed. And why would we need to use flow dialysis setup? What are the advantages, for example, compared to microfluidic devices or other techniques? Well, first of all, as I said, we need a system for continuous feed of substrates, and in particular for the continuous feed of diluted oleic acid, as well as the removal of waste products. Of course, we want to work over longer timescales. But really the key point for us is that the system, the technique that we want to use has to not be invasive. So we want to retain the vesicles, but also be able to collect them back for offline analysis, for example, by mass spectrometry, HPLC, and so on. And flow dialysis is the best technique to give us that, because there is no need for tethering, for chemical modifications of the vesicles whatsoever. We just apply them into a compartment and the filter will do the job. And also, importantly, we want to be able to sample the vesicles for the same reason that we want to be able to analyze them later on. But a system for sampling also allows us to perform instant perturbations in case we would want that. So these were the points that we had in mind for the development of flow dialysis setup that was tailored to phospholipid biosynthesis. So we looked in the literature, and there are some examples of setups for flow dialysis. Really, the simplest method that you can envision is just a dialysis bag that is sealed and is placed into a beaker with flow applied to it for constant feed. However, all of these setups had some limitations for our particular application, at least. So either the sampling was not possible, or there were very large white volumes, which is obviously not desirable, or they suffered from gravity effects. For example, the filter could be displaced or ruptured, or there could be air bubble formation, which is also very not desirable for the system. And finally, which is the example of the ultrafiltration cell, a flow is directly applied to the sample compartment. And in the case of cells, you can imagine that this is detrimental because they would be pushed towards the filter and they would therefore rupture. So the best design that we could think of is a vertical flow dialysis cell. And we elaborated on this and we tailored it to our particular needs. So after a very long optimization procedure, we came up with a modular system that is composed of one single chamber that can be then placed in an array of more chambers to increase the throughput. So a chamber is composed of two subunits, number 1 and number 2 in the picture, and each of them contains a chamber. So and there is at the interface a filter that is applied vertically. You can appreciate this better in the image below. There is a hole, the interface, and here the dialysis filter is applied. On top of the chamber is a pierceable cap that allows to perform sampling and injection. And this happens via 2 syringes, as is shown here. So again, we want to be very mindful that we don't introduce air bubbles at any time. And this is so important because air bubbles will create a volume change in the chamber that is detrimental to the measurements. So every time that a sample is taken, an equal volume should be injected into the cell. And number 6 and 11 here indicate the inlet and the outlet of flow, respectively. And you can hopefully appreciate how the flow is parallel to the vertical filter, so that we are really minimizing the effects on the filter to be as little invasive as we can. And finally, there is also a system for water circulation that allows us to control the temperature. So I want to perhaps emphasize here that there are quite some flows going around. It is not so straightforward. So the feed flow is pumped by means of a peristaltic pump to the chambers, and then the waste is collected into waste collection beakers, and you have the option to also recycle the waste, and you can do so if your feed volume is so large that you can assume the waste is identical to the feed, so that it can go through the cells more times. What is also very important is to point out that we also developed a platform to really fix all these components, and this is very important for the tubing system because it needs to be really carefully aligned in order to minimize the variation in flow rates to all the chambers. So yeah, as I said, this the development of this technology took quite some time. And what we needed to do next was to validate it, to make sure that it was useful for our synthetic cell work. And for that, the first very important control that we had to take was to ensure that the vesicles would be retained by the system. And the reason why I point this out is that we work with large unilamellar vesicles that have an average diameter of 170 nanometers. So we prepared them by extruding them through 400 nanometers. However, they come in a size distribution, and actually many vesicles have a smaller diameter than what theoretically expected. So because the polycarbonate filter that we place at the interface in the flow dialysis chamber has a diameter of 50 nanometers, we want to really prove that the vesicles are retained. And for that reason, we applied vesicles to the chamber and we sampled them over time while applying a flow to the chamber. And luckily, we observed that all the phospholipids that compose the vesicles are constant over time, or at least over 24 hours. So we could really conclude that the vesicles are significantly retained by the flow dialysis device. And next, we wanted to prove that the metabolites can equilibrate through the dialysis filter at a reasonable rate. And for that, we added arginine to the substrate feed, and we took samples from the vesicle compartment to observe arginine buildup. And in a complementary experiment, we added ornithine, so a byproduct, to the vesicle compartment, and we looked at how quickly it would be washed away into the feed. And what you can nicely observe here is that arginine and ornithine equilibrate with the same rate, and within 6 hours, equilibration occurs, I would say. So finally, a more interesting experiment, we prepared the liposomes with our ATP generating pathway coupled to glycerol kinase for glycerol 3-phosphate formation. And we applied it to the flow device. And we started ATP synthesis by addition of arginine to the substrate, to the feed flow, and glycerol 3-phosphate synthesis by addition of glycerol always to the feed. And what we could observe in a preliminary experiment was that in the absence of glycerol kinase, we could observe an ATP buildup relative to ADP over time, because ATP was produced but not used up. While in the case where we added glycerol kinase to the system, we didn't manage to observe any buildup because the hydrolysis of ATP was so fast that at no point in time we were able to obtain a buildup. So with this, I want to come to my conclusions. I have shown you our steps towards sustainable phospholipid biosynthesis in synthetic cells. And these include the development of a long-term conservation system for metabolic, of a system for long-term conservation of the metabolic energy, nucleotide homeostasis, and in particular CTP and ATP, and synthesis of the head group of phospholipids, glycerol 3-phosphate. And on top of that, the development of a technology for the control of the external medium by flow dialysis. And what is next? We definitely need to build up on what we have done so far by coupling phospholipid biosynthesis with ATP generation or import. We probably need to add a lipid scramblase to the system in order to make sure that both leaflets will expand in a similar fashion, and that we are not only expanding the internal leaflet, because this would also be disruptive for the synthetic cells. We probably will need to also add an importer of inorganic phosphate. I didn't touch on this, but we believe that inorganic phosphate will become limiting at some point in time. And definitely the final step would be to be able to perform phospholipid biosynthesis starting from fatty acids that are fed in a diluted fashion in our flow dialysis device. With this, I would like to come to the acknowledgements. I want to thank Bert Polman and Arnold Drissen, my supervisors from the biochemistry and molecular microbiology departments, Martin Exterkarte, who did a great deal of work, also together with Jelmer Conradi, and many other members of my lab, Laura Heinen, Henrik Sikkema, Wojtek Smihil, and Pauline Lefrancois, for the feedback on my work, and in particular on the flow device, and all of you for your attention. Thank you.
Thank you so much. There is a lot of questions in chat. Can you see them?
I will have a look.
If you could read the question before you answer,
yeah.
That would be helpful for people watching the recording.
Okay, higher order question. Why do we need to synthesize phospholipids? Can't we just deliver them from the outside? Well, this is a very good question. I think, yeah, but then if that would be the case, we would need to have a good system for delivery, because to my knowledge, phospholipids don't simply jump into a membrane. They are very hydrophobic. So if I imagine that we would deliver them in form of phospholipid vesicles, then we would need to have a system where these fuse with our vesicles in a controlled fashion. And I think that this would be an option if this could be achieved. But I think the scenario where the phospholipids would be added to the solution and just come to the vesicles would not work out, because they're very highly hydrophobic, and they really are rearranged into supramolecular structures. And then I don't know if this answers, but the next question would then be, how stable is CoA even if recycled? This I am not sure about. I think it was stable in the experiments that were performed by Martin, but I would not know if it would last many days or weeks. Did you express the CDSA and other membranes enzymes in transcription translation, or are those purified enzymes added to the vesicles? In our case, these are purified enzymes added to the vesicles. There is work from other groups where these enzymes are also expressed starting from a plasmid. But yeah, for that you could check the Danelon work, because he really exploits the pure system to produce transcripts and then translate all these proteins into from inside giant unilamellar vesicles. So, but we prefer to work with purified enzymes because we we have the so what we want to achieve is that we can control the system better, and we can make sure they're really correctly inserted into the membrane, because to date we do not have a system for insertion. So reconstitution is to us the best approach. So I'm not sure if I'm answering in a satisfactory way.
If someone has a follow-up question, you can unmute yourself and ask too.
And then I have, how do you control the directionality of insertion of the membrane proteins, like for ATP generation? So the directionality is not controlled, and to our experience, they either insert in both directions indeed, or yeah, some proteins, but not specifically for what I've presented, could also have a preference. However, the directionality is controlled in terms of substrates, because only the proteins that are inserted in the direction that we want will be able, for example, to take up the arginine and so on. This is great work. Thank you for presenting. When you feed oleic acid to phospholipid vesicles containing cell-free expression system, how do you prevent aldehyde precipitation in presence of divalent cations? Yeah, this is a good point. This is something that we are working on. So okay, so if I read the question better, there is no cell-free expression system in our setup. So, but we do have some magnesium around for the enzymatic activity of the enzymes. So so far we only we only have provided very diluted oleic acid over time. So the idea is also with the flow device that there will never be a build-up of oleic acid. So this will never be a problem in the future, hopefully. I'm not sure if I'm answering, but we don't have a cell-free expression system in this setup. How long can you get the nucleotide synthesis going? Even with dialysis, is it measured in days or weeks? So we have not reached that point yet, but I think we should be able to achieve it over days at least. But this has not been done yet. Is there a pathway for cholesterol synthesis too, to make membranes more fluid? We have not looked into this, but if that would be needed, then I think we could look into how we could make it. But on the other hand, I'm not sure if cholesterol would be really essential. We should first make sure that we really need to have cholesterol. Perhaps we can vary the fluidity of the membranes just by its composition. So perhaps how many bilayer versus non-bilayer forming phospholipids are produced. But in case cholesterol would be needed, then this is a new problem that should be tackled by itself, I think. This is fascinating work. Thank you for presenting, and congratulations on getting those results. Are the proteins you're using bacterial or other origin? They're bacterial. Yes, all bacterial. They come either from E. coli or lactis. Wonderful seminar, thank you. My question is, could you speculate what is the minimal number of enzymes to have complete lipid biosynthesis pathway in synthetic cell? Yeah, good question. So if we would just restrict ourselves to, maybe I could go back to the slide, to PE and PG biosynthesis, then I cannot use my computer anymore. I'm sorry. Okay, it's not working. Anyways, it doesn't matter. If we would only restrict ourselves to PE and PG biosynthesis, then we would need a set of 8 or 9 enzymes. To have complete lipid biosynthesis, and this is just the pathway that was developed. Plus, on top of that, we would need the enzymes for ATP generation, that is 3 cytosolic enzymes, plus the antiporter, plus we would need to have a system for so the system that I've shown here for CMP relation, that is 2 enzymes plus 1 for glycerol 3-phosphate formation, plus there are more components that we would need to recycle. So for example, ATP is hydrolyzed into AMP, so also an enzyme to recycle AMP there, which would be the adenylate kinase. And then we would definitely need to take care of the pyrophosphate that is formed, for example, by means of the pyrophosphatase. Later on, a serine is needed for phosphatidyl ethanolamine synthesis, so perhaps a serine importer or so on. As I mentioned earlier, an inorganic phosphate importer, as well as a lipid scramblase. So it is quite a complex system. I didn't count all of them, but this is what we speculate is really the minimal number of enzymes. And of course, we could make it a little bit simpler by replacing the arginine deiminase pathway by means of an antiporter. So for example, exploiting the ATP-ADP carrier protein from the mitochondria, this would be a very fast antiporter that would directly import ATP from the external medium. So that is maybe cutoff that we can also keep in mind. But it is a very, very complicated system. Thanks for answering my question. Congratulations on great work. But I think I've answered all questions. I'm not sure if there are more.
Thank you so much. I think, yes, you answered all the questions. I have my own kind of more of a speculative question. Do you think all those enzymes that you listed, will we be able to express all of them correctly in TXDL, or will we always need to add at least some of those really tricky membrane proteins from the outside?
Yeah, exactly. This is a very good question. And perhaps you're, it's really a question of how efficient the transcription and translation system will be in the future, right? Because at the moment, I think it will not really be feasible, but it is difficult to foresee it will become so efficient that we will really be able to express all this protein. But for sure, if we are aiming for a synthetic cell, we should be able to express far many more proteins than just this for phospholipid biosynthesis, right? So all other or other proteins that are. So if we are aiming for that, then the answer would be yes. But maybe on the road to there, then we would still need to cheat and make the synthetic cell a bit or feed it from the outside a little bit more.
Yeah, I think so too. All right, thank you very much. If there are no more questions, I want to thank you, Eleonora, again, and thanks everyone else.
Thank you for hosting me.
My pleasure, and see you all next week.
Thank
Bye.
you. Bye-bye.